And I'm very happy to announce the next wild guy here. This is Fabian Theis. He's one of Europe's leading researchers in AI and health. He's a professor at Technical University of Munich and director of the Helmholtz AI Center. And he's leading cutting-edge research in AI-driven biology and medicine. He's a Leibniz Prize laureate, chair of the Bavarian AI Council, shaping AI strategy at the interface of science, medicine, and policy. And beyond all titles, he's a great collaborator and guy to work and be with. Please give a hand to Fabian Theis.
Thanks so much. Thanks, Michael, for the kind introduction. Thanks, Minister Blume, for coming in a very wild form. This wild thing is not going to go away, I guess. And yeah, thanks everyone for interest. AI is here, right? You heard AI is the language how we model biology and its perturbation medicine. So this is working out. But what I want to ask today is, what's the impact? You know, how do we measure that impact? We build great models, and all of us sort of making benchmarks, and the accuracy is there. But what actually matters is not that, but you know, it's the impact in terms of years of life. This is a measure. It's called daily disability-adjusted life years. It sort of really basically quantifies. Hate to make it a bit sad now, but this is sort of what we're going to die from, right? This is the big overview of. And this is a global picture. There's some sort of local versions there as well. But the big sort of killers are obviously, you know, cardiovascular type of diseases, diabetes increasingly. This is sort of all the chronic part. This is injury, and this is sort of communicable diseases, or stuff that you can sort of treat, infectious diseases and so on right away. So this is where we should be looking. And very often, when we design things, we look maybe for things that are easier to address. So let me maybe just briefly sort of show you once more these statistics, because I think it's so fascinating. You know, if you want to sort of pick your new research topic or maybe a new area to fund, you know, here you see sort of what's dark is sort of getting worse over time, and you see cancers and so on, they start getting treated, but some of these sort of big killers, neurological diseases and so on, this is stuff where there's also not so much medicines yet available. Particularly in that field, you know, things have been stagnating for 20 years. So there's a real need for new medicines, but this stuff is complicated. So let me maybe sort of give a bit more examples. So what are the big percentages covering the thing? And by the way, you know, if you look for Europe, but Germany in particular, this thing is shifting a bit over there. So apparently road injuries, we're getting better. We still have some falls. Sorry for that joke again. but I think there's a few reasons why we're missing proper treatments for that. So here, for example, stroke, cardiovascular, kidney diseases, diabetes, lung diseases, right? These things are complex, chronic things that sort of develop over a longer time span, and you need to find ways how to sort of really continuously treat this. It's typically multifactorial disease, so single drug, single treatment just doesn't work. You need combinatorial stuff. Similarly, for neurological diseases, also neurodegenerative diseases, these things are not even easy to quantify. Like, when do you actually start to have Alzheimer's? Do we have sort of molecular things? It's always a phenotypical thing. So bringing this to a molecular thing where you then actually want to come up with disease treatments is hard. This is a reason why for these things we just don't have so much stuff. You know, obviously it's not all bad, right? For a lot of these communicable diseases, there's very strong treatments because it's like one virus, one bacteria, and you can sort of do things against that, right? But I think it's safe to say that where we have the highest burden, which is visualized here, typically is not where the easiest biology lies, right? So we need to come up with ways now how to find how we can treat this. So I think and this is quite natural, right? And I'm talking about pharma, but I'm talking also about scientists. What we optimize for is not the disease burden very often, but it's where we have sense, what's sort of experimentally convenient. So my point of this whole lecture kind of is, and I want to be really short, but I think the key point is that we do have data. But in biology, data is not found, it's created. We need to sort of measure it, we need to make it available as well, right? So what's the optimal data in biology and in this perturbation medicine, what we should be creating? Biology is not really data limited. We can do so much, but it's decision limited in the sense that we need to think about properly what the best data is that we need to feed to our AI model. Not just what we understand, but what the model actually needs to really optimally build it. We have been building foundation models quite successfully in my lab, for example, now for spatial biology, really scaling up, sort of hoovering up all the data that we have around. Still, they typically give not the most new answers, but often answers to questions of yesterday. So we need to find new ways how we address this. So if we just keep the same experiments faster, we might not get there. So the typical answer is, in AI, what do? We just sort of scale, right? But if we scale with sort of just looking again and again what sort of typical, like what sort of the field is looking at, we might not ask the right questions, right? So scaling is not cutting the thing. In fact, actually, drug development, I think something like 90% of clinical trials fail. So doing AI-guided trials is something that really people look a lot for, and we need to sort of find ways how to go there. Effect size is often very modest of the found treatments. They just sort of address a subpart of the population. And also these complex diseases just don't need a single, but they need sort of combinatorial complex, potentially also adjusted and personalized interventions. So I think it's safe to say the failure is not in the models. We're getting better at that. I mean, sort of really work with the general AI community, but I think the experimental strategy, this is missing, not the intelligence. So the question that I want to ask is, and I think this is a question that's going to be massively driving, in general, AI in the sciences, but in particular, in one of the sort of most needed fields, where we just don't have big models. The question I want to ask is, how do we decide which experiment to run next? If we are able to answer that properly, we can guide, we can sort of have our little GPT that helps us now guide, like writing emails or whatever, right? Trying to help us, what is the next type of thing to do to make our models even better, to find the better treatment, better diagnosis, and so on. Let me before answering that, so just really be concrete and show you where this already works. And this is a domain that I like a lot. Disease happens on the cellular level. So let's look at cells. We're just flying through a big data set of cells measured on the RNA profile, sort of grouped by similarity. And what we essentially build is sort of descriptions of where we are. We call these atlases. In these atlases, we very well know, this is sort of what's understood, but we don't know yet where to move. Right? But if you look sort of on the map, maybe you can see where you should be going. But how we move there, this would be the experiment. So let's build first these atlases. And this is actually what we've been doing quite successfully. This thing is called the Human Cell Atlas, an international network of more than 1,000 labs, sort of spiritual successor to the Human Genome Project. And across the whole body, we're building sort of descriptions of what is. Not yet where we go, this would be the experiments, but just let's describe where we are. Right? And for example, my lab has been building the first integrated, and this is sort of an foundation integration type of thing for the lung. We've been extending this then also for the pancreas. This is where diabetes happens, very exciting, but also a lot of cancer types we can sort of map there. We've been recently doing this for the human retina, sort of as a first entry point to the brain, and also for brain-derived organoids. These are sort of replica of the brain where you can actually then do screens at scale. Very exciting, very useful to sort of help design the next experiments. But again, this is sort of not answering the question what we should do next, like what we should ask our experimental partners to do, right? So they tell us where we are, not how to move. So how do we do that? So this is, I think, a really pivotal point that I think 2026 could potentially get. Last year was the year of the agents. Let's now put the agents to work, right? So let's get them do something, right? So I think this idea of generative modeling, predicting what the effect of a perturbation would be, I think is exciting. This is from a perspective we did with Yosha Benja the other day, and the idea is essentially that you have these perturbation experiments and you sort of see what type of different things you can do, what type of potential effect they have in a cell, indeed also combinatorial ones. We just actually set up a startup called RN AI. Nice, right? Where we sort of do these combination treatments. But I think the idea is that we kind of want to model these combinatorial treatments, and we want to sort of see what they should be doing. And this idea, sort of having such a virtual model, is nowadays sort of often coined virtual cell, there's a lot of excitement about that, right? And for example, Demis just recently, actually quite some time ago, when sort of they came up with AlphaFold, said, we want to build a bottom-up virtual cell model. Or Priscilla Chan and Mark Zuckerberg and the big sort of CZI Foundation have been pushing this idea of virtual cell modeling for quite some time. So I think it's a very exciting area where we could be sort of building on top of that. And my lab has been building these sort of forward models. Recently, we call this thing cell flow. We have sort of adapted versions before, where we can now tell you when we sit at this point in an atlas, in the description, where we could be going. But this does not yet solve this question, hey, what should I do in terms of data? So the typical thing, and this is sort of what academia very often does, but also I think many sort of analysis, data science type of things. We have some type of data, we build a model, and then we get some insight, and we tend to stop there. So the new kid on the block obviously is putting what's called the lab in the loop, right? So we do these cycles, so we sort of iteratively improve on our models. It's like in language models, right? Where we saw, hey, let's do some reinforcement learning to sort of really get better at iterating on these things. And what I would say, what this means here is that once we have the model, we now ask the model, hey, where are you already good? Where are you sort of missing information? It's an interesting strategy. It's often called exploitation versus exploration. So early on, you want your model to sort of see what is missing in the space, but then once you have it, you sort of drill down and sort of get better and better, help me really understand if it's this particular protein pocket or something else, to really improve upon it. So these trade-offs, very exciting, a lot of science and sort of modeling happening. But if you crack that, right, you're going to ask in this whole space of, and we're going to see this in a podium discussion, I mean, this whole space of protein design, one of the hottest things, just got the Nobel, right? Like, what's the real question to search for there? So I think a lot of excitement here. Let me just show you two brief examples from that. And I won't want to get very technical, but this is a paper that just came out that we brought out in Science last summer together with a company from Boston, where essentially we asked, if we know for drugs what they do in terms of sort of signatures, gene expression signatures, can we go, and there's some big databases for that, can we sort of weight and rank them with respect to some type of phenotype they look for? In this case, maybe killing a set of cells or sort of enriching a certain population. And we came up with a very simple model, just a multi-layer perceptron, very simple neural network that did a bit better than others. This is sort of quite straightforward 101 AI type of thing. So that was nice. But the novelty came in there, that we not just now sort of went with the model and stopped it, like, this three-stage approach, but that we now took this one, we made some experiments that were sort of asked by the model, and then we sort of looped it back. So we had this closed loop type of active learning. And then we sort of came up with a signature and we improved upon it. And then, we sort of had our validated lead compound. And I think what's interesting here, and there's really money in that, right? It's not just sort of interesting science. We could show that versus a random baseline, you sort of increase at least tenfold. So you can either measure 10 times or 13 times as many drugs as you could before, or you sort of just save and go down to 10% of the cost. And this is, I think, the acceleration that we expect from AI to happen, right? To really sort of amplify and speed up our processes. This was sort of a very simple model. We have some more complex ones. And again, just excuse me the technical sketches, but I'm super excited about this. This is all unpublished, so I just want to sort of, I don't want to get too technical, but maybe whet your appetite a bit about this idea. So, we have like these spaces. This is sort of where we move across. Remember, I said we had these atlases, and you want to sort of take steps in the directions that, the next drug should be selected. And we can essentially map from this one to the observation space. And this would be the forward models, and now we have these inverse models based on an idea called flow matching. This is sort of, at last NeurIPS conference, like this biggest machine learning conference, half of the talks were about this idea of flow matching, where we can sort of iterate between things. We can now ask, hey, I want to go there. What type of experiment do I need, right? And this is what we can do. And I have just one short example, and I don't even properly explain. I just want to make the key point. With this, we can not only do drugs, but we can do the next thing called cell therapy. So we can ask our experimental partners to just take fibroblasts, just skin cells, and throw a whole bunch of what's called transcription factors. This is sort of what the body uses to sort of program cells. But these things are combinatorial, right? So we throw 50 on top of these, but then we would need to go through like 32 billion or trillion combinations on it. This is something that we can't even do in the computer. But because we have this search engine now, like we have this inverse model that allows us to automatically search for this. We can now come up with, in this case, we want to make cells that kill cancer called dendritic cells, or could we sort of want to really specifically engineer those? And then we can go through our model, ask it, hey, which sort of cocktail combination do I need to put on top of that to make it? And then we find a few that really sort of look like this picture up there. So basically, the key point is these colors here look a bit like up there, and we can quantify this, and this is all working out. So this is ongoing. This is not yet published, but I'm really excited about basically a systematic way how we now not just sit down with some of our experimental partners, analyze the data, and write a paper, but we go back to them and say, guys, let's use these 3, 4, whatever transcription factors, and you can make the new cells that you can get. All right, so taking a step back and wrapping, what I showed you is that we have ways in the cellular space to explore not only what will happen if we make an action, but what also allows us to select the best optimal action, right? So this learning, we have colleagues from robotics here, they're used to that. You know, like when a robot needs to go through a maze, you need to sort of plan some strategy. And we can now do the same thing for science. I showed you an example here for cell sciences. We can do the same thing in large-scale medicine, but you can do this also across all of AI science. And I showed you this example here just for the cell level, but of course the human is a little bit more complex, right? So we go up to tissues, we go up to the organ and the full human level, and what the key challenge is now is sort of to bring these scales together. And what I'm very happy is here with really generous funding from the high-tech agenda from Bavaria at our place here at the Computational Health Center in Munich, we've been bringing together more than 600 scientists, 45 PIs from all over the world to really build these joint models. And what we do is now we're intersecting them. We're calling this not just a virtual cell, but the virtual human, and essentially have on each of these levels models that we now can pull together in the sort of a big picture that not only allows us to describe the state where we move, but, and I think this is really cool, now for each of those scales allow us to ask the question what we should do next. So adapted clinical trials, this type of thing, this is what's happening here in Bavaria. This is, I think, where a lot of sort of value generation is happening. And yeah, I look forward to a discussion maybe later here now at DLD or in the upcoming panel, and I thank you very much for your interest.